Hi there, welcome back to this video on autosomal linkage, where we'll be continuing to look at crossing over and the variation that introduces. So this is what we looked at last time, and we want to pay attention to what happens here at meiosis, where we have the sister chromatids lining up and generating the gametes that are available to create the 3:1 ratio here. So let's move on then. I'm going to simplify my drawings a little bit so it's easier to see what's going on, but let's go with this. So we start off with R and G on the same chromosome, and we've got a homologous pair looking something like that. Now I'm going to make it simpler than that. So during meiosis, the chromosomes duplicate to create a set of chromatids, and I'm going to use green here for the homologous pair. So they're lined up during meiosis, something like that. Now if they continue to go through to create the gametes, we're going to end up with a 3:1 ratio. But let's see if we can demonstrate crossing over here. So when they're lined up close in the during metaphase, we've got the left hand chromosome starting to look like this, and let's have the right one doing that. So these inside chromatids are able to cross over. Now I'm going to make this simple and have it cross in one place. But it can happen in multiple places and create lots of variation. So let's make them actually cross over the top of each other. Alright, so there's the breakpoint that we're interested in. And I'm going to stop that chromatid there. Let's do the green side and make that one stop there, but then continue that one with green. And there's the purple. Now let's separate them out. Alright, so now we've had the crossing over event that has led to whatever genes are on here, and their alleles on this section are now part of the other chromatids from the other set of the homologous pair. Alright, so if we start off with big R, big G, big R, big G after the duplication, and we have little r, little g, and little g. After crossing over occurs, the outside chromatid remains the same, so we've got big R and big G, big R there. However, the big G from this part has now moved to the other chromosome, and we have little r, little g on the outside, little r, and little g is now on the left hand chromosome. So if we look back again to our combinations that occur from normal meiosis, we have big R, big G, big R, big G. Let's stick with the green here. Little r, little g, little r, little g. So those are our gametes. But now we have big R, big G, big R, little g, little r, big G, and little r, little g. And just going to make these all purple gametes. So the crossing over event has led to the production of 2 new gametes, and this introduces extra variation. So let's do a cross between an individual who produces these gametes and one where no crossing overs has occurred to see what the possible variation looks like in the outcome of the cross. So let's say we have a male and with gametes of big R and big G and little r, little g. So no crossing over has occurred there. And female crossing over has occurred to produce the extra gametes. So big R, big G, big R, little g, little r, big G, and little r, little g. Not the tidiest work, but hey, not going to use a ruler in an exam. Big R, big R, big G, big G, big R, big R, big G, little g, big R, little r, big G, big G, big R, little r, big G, little g. And here, big R, big G, little g, big R, little g, little g, little r, big G, little g, and the homozygous. Alright, so what do those look like? So just to remind, big R equals round here in this example, little r equals wrinkled, big G equals green, and little g equals yellow. So let's figure out our possible phenotypes. So offspring phenotypes. Equals round and green, and we've got one of those, 2, 3, 4, 5. And we've got round and yellow, and we've got one of those times 1. Do we have any wrinkled? Yes, we do. Let's see if we've got wrinkled green, and we'll look for wrinkled yellow. So we need the round and green. Let's just check those. So round and green, there's 1, 2, 3, 4, 5. Alright, do we have round and yellow? There's dominant for round and our homozygous recessive for yellow, one of those. Wrinkled and green, we need recessive little r's, there we go, and dominant for green, so we've got one of those, and homozygous recessive wrinkled yellow. So now, instead of having the ratio of 3 to 1 that we expect from having no crossing over, we're ending up with 5 to 1 to 1 to 1 in this example. So that's without considering other types of crossing over. Now, the final part of crossing over is that, let me just draw one of our chromosomes. So we have a gene here and a gene here. So that was big G, little g. And in our previous examples, we were doing something like that, quite far away. Now draw that again, but have the genes closer together. So we have big R and little r there. Now, when I drew the crossing over event before, it was down here. And you can see the further the genes are away, physically in distance, the more likely crossing over will separate them to create the extra variation that we're seeing here. But if they're close together, they're more likely to be inherited together. So we keep, even though a crossing over event is occurring down here, these two are kept together on the same chromosome and reduces the variation. So that's one other thing to consider with crossing over. All right, a little bit complicated, but we got there.